record on this computer. Recording in progress. Louis, just so I'm from a small town in Pennsylvania, so I don't massacre your name. Can you just tell me how it is, how you say it?
My last name is pronounced Garegnani.
Okay, I'll probably massacre that a little bit, but
don't worry, I'm used to it.
Okay. All right. So everything is up. So thank you both for joining today. And this is the Trial Site News podcast. I'm your host, Dr. Erin Stair, but please just call me Erin. And we're going to talk about ivermectin today, which is obviously always in the news. There's always a controversy. As it stands now, we know that the FDA doesn't recommend it for COVID-19. The World Health Organization doesn't, and the NIH doesn't recommend it, and they don't not recommend it. They kind of take a neutral approach. So today we have Dr. Louis Garegnani. Thank you for joining us from Argentina, and Dr. Pierre Kory. Thank you for joining us as well. It's going to be great to hear from you both. So I hope we can have kind of a really friendly, informative discussion, hear both of your guys' perspective on this. So to start it off, I wanted to actually pose my first question to you, Louis, if that's okay. And so I read your opinion piece in the BMJ Journals that you wrote in February 2021, and that was titled Misleading Clinical Evidence and Systemic Reviews on Ivermectin for COVID-19, in which you wrote, evidence and research related to ivermectin in COVID-19 has serious methodological limitations, resulting in very low certainty of the evidence. So my first question is that still your opinion? And can you explain why or why not?
Yes, I think I still think that certainty of the evidence related to ivermectin it's still low. And although there are, since we wrote that paper, some new randomized controlled trials arose and were published in several journals. Those that are methodologically well conducted, at least from an external point of view, with low risk of bias and prospectively registered, with a great reported allocation or randomization process, they didn't find any benefit of including ivermectin for COVID-19. So, and I recently checked the World Health Organization recommendations and the Pan American Health Organization recommendations, and they stand the same. Nothing has changed since February, just that we have more trials. And so that's basically
so it. You stand by your original position? Okay.
Yes, I think, please don't get me wrong. I'm not saying that ivermectin is no good for COVID. Yeah. But I think that the available evidence has serious limitations, and it's probably a good field for research.
Okay, Dr. Kory, can I give you a few minutes to respond to that?
Yeah, I mean, so we have very different opinions, right? So when you look at the existing randomized controlled trial evidence base, you're talking about 26 randomized controlled treatment trials. Just on treatment, you have 26. I don't know when the editorial was written, but it seems quite outdated. And even for the time, my challenge when I see sort of editorials and reviews and comments on the evidence base is that, and I think Luis would admit, that in order to really judge the quality of the evidence, you need really granular details of the trials, how they're conducted, like you said, allocation, concealment, follow-up, randomization schemes. And the challenge that I find is those panelists who have really delved deep into the evidence base and amassed all that data, and there's a few groups around the world who have done that, and those are the ones that I would listen to, meaning that I would put the most importance on. So one group in particular was hired by the WHO and Unitaid, and that was led by Dr. Andy Hill. And it was his work in which he identified all of the active trialists in the world, 59 active randomized controlled trials. He identified them in January, and he's been following along, and he did a lot of that work. And his last public lectures And the ones that he shared with me, he actually, after finding out and communicating with all the investigators, he grades the evidence as moderate certainty. And I have to be really emphatic about this, because although this is an important debate, but there are lives at stake. Ivermectin, the moderate certainty evidence is on mortality, which is the most important patient outcome you can have. So we have a drug that has been identified by not only the expert lead researcher for Untitled and WHO, but also an independent group called BIRD in the UK, who has done similar work, independently reviewed all of the randomized controlled trials. They also grade for the outcome of mortality the evidence to be moderate. They have delved deep into all of these trials, amassed them. And so you have two independent panels who've done systematic reviews, meta-analyses, and grade the evidence as moderate. And I have to remind everyone that corticosteroids, which are the standard of care worldwide, have graded the evidence as moderate. Now I'll stop there. So, and Luis, and the other thing that we're going to differ on is, I think Luis is really probably referring to the Lopez Medina trial published in JAMA.
I'm going to get there, Dr. Kory.
Yeah,
that's the whole
I just
thing.
want to say that that's probably its own debate. I
Yeah.
just want to say very briefly that the opinion that that has a low risk of bias is not shared.
We're going to get there. I promise you. I just want to make sure we move along. So the next question, and we could start with you, Dr. Kory. The idea, as I understand it, when sort of like this aha moment, like ivermectin might work against COVID-19, came from in vitro studies showing that it could inhibit the virus. And people were very excited about that. But then a lot of people who were doubting that said, well, you're going to need 9 to 10 times the FDA approved dosing for that to work. On the FDA's website, they state taking large doses can cause serious harm. Dr. Kory, I know you and the doctors from Frontline COVID-19 Critical Care Alliance, you developed a protocol. So how can you assure people that that dosing is correct? How can you validate it? How will we know that it's safe?
Yeah. So a couple of things on that. So that is a very common sort of objection to ivermectin, and it's very poorly founded. So number one, all around the world, you see all these agencies, and even in Luis's editorial, he mentioned that really you shouldn't even consider ivermectin as a therapeutic because it can't attain the dosing concentration used in a cell culture model in Australia. A couple of really quick things. Number one, cell culture models in humans, two different things. Very well known that they are oftentimes not transferable. So the doses used in a cell culture and the dose used in humans are different. Number two, that was a monkey kidney cell model. Kidney cells, okay? There's not a lot of high concentrations of kidney cells. When they've repeated the experiment in alveolar lung cells, and again, you need to be an expert at the disease, not just an expert at evidence-based medicine. Be expert at ivermectin. This is all we do is eat, sleep, drink, and breathe ivermectin because we're trying to save lives. When you talk to Kelly and Wagstaff, they've repeated those experiments. This is personal communication. Their article is under peer review. But they have achieved inhibitory concentrations in lung alveolar cell models easily, number one. Number two, the doses in tissue, in adipose and lung tissue are 3 to 10 times higher than what you'll see in serum. So you're getting much higher doses of concentration in tissue. And then lastly, is that if indeed you could not achieve effective concentrations, we wouldn't be sitting here today. We wouldn't be debating all of the efficacy in all of these trials. I mean, to say that they're methodologically limited, that's one thing. But what I've never heard anyone say is proof it doesn't work. All they say is that the proof that it works is suspect and methodologically limited. Fine, have it be suspect and methodology. However, it's there, it's repeated, and it's consistent. So to say that you can't achieve a concentration, it just bears no relation to reality, what we know about the science of achieving concentrations and efficacy.
Thank you. Dr. Garegnani, do you think ivermectin is a safe drug, or do you have concerns about adverse effects?
Well, that there is, I don't remember any evidence related to the use of ivermectin and adverse events here in COVID. So that is one thing, but I'm really concerned that we are trying to say that ivermectin has some clinical benefit based on the effect on cells. So we are trying to translate the effect that ivermectin may have in an in vitro environment to the clinical setting. So that is at least questionable. Yes.
Yeah, so can I just agree with Luis? I agree. I always say this, very few things make the leap from the bench to the bedside, right? So stuff can work well in the lab, it looks good, but when you bring it to humans, it doesn't really work. But I'm actually none of my arguments ever rest on the cell culture model. I'm a clinician researcher. I really know what it's like, out in the real world. I look at clinical evidence, and that to me is the one that's unassailable. I agree. The bench is not really what's going to save a life. That's a cell culture.
Oh, go ahead. Did you want to say something?
I just wanted to add one more thing, that we have a large variability in trials using ivermectin. Population where receiving ivermectin is highly variable. There were close contacts of positive COVID patients. There were mild to severe or moderate COVID cases also receiving ivermectin. Doses were highly variable. The routes of administration were highly variable. They were already taken. Now I saw a trial of aerosolized ivermectin or spray ivermectin. So outcomes, definition of outcomes, in order to say that ivermectin has any impact on health, they were also highly variable. So I'm not saying that ivermectin is not working, but we should at least take a good definition for outcome, for population, for doses, and try them in randomized settings.
Okay, and just to add a comment, because I did look this up for the listeners, there was a systematic review published in 2020 in the Journal of Antimicrobial Chemotherapy that revealed no difference in the number of individuals experiencing adverse events between standard and higher doses of ivermectin. I think one trial showed ocular events, but the FDA on their website has said they've received multiple reports of patients requiring medical support for self-medicating with ivermectin for horses, which is probably something nobody should do. Anyways.
So, Erin, can I comment on that? So, first of all, the safety, I would like to just say that's absurd. That's a drug that's been given 4 billion times in mass distribution throughout huge portions of the world. Many papers showing overdoses with ill effects. In fact, one of the world's famous toxicologists reviewed 350 articles and recently issued a report. And in his summary, he said, severe adverse events are unequivocally and exceedingly rare. Even in all of the trials in COVID, you can find no harm signal. And just last week, a report out of Argentina of 3,000 patients using very high doses, actually. I was actually concerned. They gave 0.6 milligrams per kilogram for 5 days. And of the 3,000, not one severe adverse event was observed. And so the safety for me is unquestioned. But what's interesting about Luis's summary, which is totally accurate, right? So he's saying that different populations were studied, different doses, different routes of administration, which is not really true. I mean, it was almost all oral. Yes, there was one kind of nasal spray, but you're generally dealing the entire evidence base was oral administration. But here's the thing, that seems to me as the classic glass half full, glass half empty, right? So whereas he looks upon that as not being able to establish a rigorous effect or guidance on how to use it, I actually look at it differently. I look at this massive database of literally, if you can consider the prophylaxis trials, we have 36 randomized control trials, 60 control trials. That's a huge amount of patients. And when you separate it out into those buckets of preventative, early, and late, and then the doses, it gives you a wealth of information. Keep in mind, when you do one large trial, it tends to be on one population at one time. And so you're left with an evidence, let's say you have an evidence of benefit. What you're immediately left with is lots of questions. Well, can I use it earlier? Can I use it later? Can I use it in a higher dose or a lower dose? Well, when you have such a variety and diversity of the evidence base that you have now, you answer a lot of questions, which is that it works as a preventative, it works early, and it works late. The other thing that's really critical is that the Untitled WHO researcher has lectured on and reported a dose-response relationship in terms of times to viral clearance. That is a major scientific pillar of efficacy when you see a dose response. Single day versus multi-day dosing and higher versus lower, you see a consistent signal to the times to viral clearance are faster. And so I have to say, to me, that diversity of trials and doses and context is an incredible strength of the evidence base. And I tell you, I'm really grateful for it because it gives us really good guidance on how to use the drug.
Did you want to respond to that at all, Louis?
Yes, there's another issue with that diversity of trials, that they are pulled all together in those meta-analyses that Erin has mentioned before. And that is at least methodologically questionable, because the underlying clinical question beneath those meta-analyses is highly heterogenic. So if you mixed randomized controlled trials that use ivermectin for prevention, and you mix it with other trials that use ivermectin for treatment, the clinical or the research question underlying those two groups of trials are different. So you shouldn't pull those together in one effect measure that a meta-analysis can do. So they should be treated in a meta-analysis separately.
Fair point.
I agree. And I don't know of any of the credible meta-analyses that lump those two together, the prevention and treatment. No one's doing that. And I agree that's methodologically would be incorrect. There's different outcomes.
Totally, and outcomes are very different too. So
Right.
To base a recommendation in those meta-analyses, that I have my concerns with that.
And I think concerns are fair, Erin, but here's where I sort of struggle with these overly, I don't want to say overly statistical methodological limitations. It's this excessive statistical caution. When you're looking at 60 trials, where 58 of them show a benefit, oftentimes large magnitude, yes, sometimes small, so you don't reach statistical significance, but almost all of them reach near statistical significance with large magnitude. I've never seen a consistency of benefit across 60 trials. And yet we are expressing excessive concerns on one of the world's safest drugs. And so my last point on when you're assessing an evidence base, yes, I agree that you want to be methodologically correct and consistent, and you want to be protocolized and prospective. However, it's hard to make a mistake on this evidence base. And the other thing, as a clinician who treats patients, every day I go to work, to borrow a famous phrase, I go to war with the evidence I have, not the evidence I want. And on this day, if I'm seeing a patient who's ill with COVID, based on the existing evidence base of this massive amount of trials, all showing consistent benefits, I'm essentially ethically and morally mandated to use it. Not to sit there and wait for the perfect systematic review, meta-analysis, which carves it into these nice little buckets. I can't do that. As a clinician, I really have to use my best view of the evidence as it exists now. And I have to tell you, it's really unassailable to me, if you look at the totality.
Okay. So we already basically discussed the systemic reviews and kind of beat the data to a dead horse status here. So let me ask you about things that people have observed in the real world, so to speak. So some places in India, Mexico City, Peru, some people, they were handing out bags basically that had ivermectin in it, and they saw, oh, look, there's a reduction in cases, there's a reduction in mortality. And other people said, well, you didn't isolate, say that that's definitely ivermectin. It could be other public health measures such as lockdowns, for example. Would either of you like to address that?
Well, So I work pretty closely with Juan Chimie, who's probably, the most well-known analyst looking at these correlations between various infection control measures and then ivermectin use. And he's been reporting on this for over a year. And his paper, which he wrote with David Scheim and Jennifer Hibbert, it's under peer review now. They show very carefully, and they assessed all the confounders, but they show very tight temporal associations in regions of Peru that did ivermectin distribution with rapidly diminishing death rates, excess death rates, which are really the most accurate, and case counts. And he's really shown that across the world in almost every place that it's adopted it. And they carefully look at the timing and impact of lockdowns, other masking measures, Google mobility data. I mean, they were very careful, and I find it that those relations are really very, very tight. Now, let's move that aside, though, because we do have to talk about Mexico, and I think it's really important. Because the Mexican IMSS, right, the Instituto Mexicano Social Seguridad, I probably butchered that, but they're in charge of a good portion of the healthcare system in Mexico. And on December 29th, they did a nationwide test and treat program where everyone who tested positive got 2 days of ivermectin, 12 milligrams, not a high dose and not a long duration. And they just put on a preprint the results of that. So what you saw after they instituted the program is their hospitals were full, they were in crisis. And you start to see a very rapid reduction in hospitalization and death rates and case counts across the country, to the extent that now COVID is really, it looks almost like, it's almost gone. And they put the results of that program, they very carefully studied those patients, 150,000 patients that they studied. I think it was somewhere around 70 to 80,000 who took the ivermectin and maybe 50,000 that didn't. Very well matched as far as age, degree of illness, symptom duration. And they showed a massive reduction in hospitalization and death. And that's similar to Argentina, who last week also reported on a smaller program done under the auspices of the WHO. Also same thing, reduction in hospitalization, death, and ICU. And then in India, you have the states of Uttar Pradesh and Bihar and Delhi all reporting massive reductions in death and hospitalization with widespread use of ivermectin. So you have a ton of real world evidence for this.
Thank you for that, Pierre. Lewis, do you want to respond to that?
Yes, I think that you have to look at the design and how that research is being conducted. It's not prospectively registered. You don't have a protocol of that research. I know they are handling data on the go. They probably, it's like a retrospective analysis. So it shouldn't be randomized in groups. It should be treated as a cohort study, maybe. And of course, if they then or they perform an adequate control of probably effect modifiers or confounders that might appear during the research on their population, you can establish an association between the ivermectin effect and those benefits that you're saying, but you cannot prove a causal effect. So I think that those are not the minor issues. That some research has been done with no protocol registration, and that's basically why some of those results of those papers appear like preprints in different repositories, and they don't get published in like formal journals. So I think that is an issue, probably because the risk of bias in those research might be high. So, and I have my concerns of administering any treatment based on high risk of bias research.
Yeah, we have to be very careful with the statements we make. We have to make sure that they're factually correct, because to say that these trials were unregistered prospectively is actually false, right? So in the most recent review of 24 randomized controlled trials, by Brian, which is a pre-publication in about 2 days, they have evidence of at least, I think it's 20 or 22 that were registered. This is of the 24. So to say that they're unregistered trials is factually incorrect, number one. The other factual...
what happened with the other 40 trials that you mentioned just earlier before?
I'm talking about in a systematic review of just randomized controlled trials. So let's talk about observational trials. So in observation, so the other factual incorrectness, and this is really pervasive and it has to stop, which is this obsession that the only scientific truth can be extracted from randomized controlled trials. It is well known throughout history that randomized controlled trials are subject to multiple flaws, just like observational controlled trials. And when you look at the history of the two trial designs, you cannot find a credible difference in findings. They generally match throughout history. Observational controlled trials are valid. I do agree they need to be carefully done and methodologically rigorous. Many of them are. There are techniques to do that. You can do propensity score matching. And one of the largest trials in the United States, which came out of Broward County, Miami, which was published in a major medical journal. It went under extensive peer review. It had propensity score matching to a very high degree, and they reported very large mortality benefits in a hospitalized patient, even in the critically ill. So you know, I really have to say that data can be taken from many different trial designs. You can look at it in multiple different cohorts, observational, and to have this preference that it has to be randomized is wrong. I've seen randomized controlled trials get things wrong, just like I've seen observational trials.
I totally agree with you on that, but you cannot say that based on an observational study that the effect you see or the association you see or you find in an observational study is because of the ivermectin. You cannot establish, given in an observational study, you cannot prove causality, a causal effect from due to the intervention you're applying. It's an association. Maybe statistically, that is maybe what you can use for later on performing a randomized controlled trial. I totally agree with you. Some randomized controlled trials are not well conducted. You have already mentioned some randomized controlled trials on ivermectin were not well conducted. I agree with you on that. I think, but I'm not saying. I repeat, I'm not saying that there is no beneficial effect on ivermectin, but I'm really saying that I have my concerns on the issues on the studies and on the methodological designs, and we cannot provide any recommendation based on those studies.
All right. So correlation versus causation, there's a hierarchy of evidence that we follow, but I think it does have value, especially in a pandemic situation with a new virus, right?
Totally.
I mean, that's kind of what we have to do. We have to observe and see what's working. But I also agree that the highest, as we've been passed down, I know Pierre has issues with randomized controlled trial, and I would agree, especially today. But I guess proving something works, and testing it against a placebo, which I know you also have issues with doing that. But isn't that, I guess, and repeating that process and showing it works better than a placebo, as much as you can, wouldn't that convince you more? Maybe not you, Pierre, but like Luis, is that what you're saying? That's what you want to see? I
would like to see at least a well-conducted randomized trial in a predefined patient population with predefined doses. And with a correctly defined outcome.
That's actually a great segue to my next question, because this has to do with how a trial gets conducted and who funds it, right? So as far as I know, the only randomized controlled trial that was published in JAMA, which is a top medical journal, is the one that was conducted in Colombia. It had, I want to say, 476 people. I wrote down some details here, right? And what they found was there was no statistical difference between ivermectin and placebo when it came to symptom resolution. The thing that was most striking to me about that study was the funding that the authors received. And I just want to mention some of them. So several pharmaceutical companies were involved with that study. Johnson being one. We know that Johnson has one of the 3 approved COVID-19 vaccines here in the US. Gilead, which makes remdesivir, also provided funding for that study, and they have a keen interest in that early treatment market. And Merck also funded that study, or gave funding to the researchers. And we know that the US government just signed a $1.2 billion deal with Merck to purchase their new antiviral for early treatment of COVID if it proves successful. So I'll start, I guess, with you, Dr. Garegnani. Would you consider that a conflict of interest? And would you trust the results of that trial?
Almost all randomized control trials related to the pharmacy industry, the pharmaceutical industry, are industry funded. So if that might be an issue, of course, I don't see if Merck or those companies that you mentioned producing ivermectin that might have an impact on the conflict of interest or on the funding related issues. It's something that systematic reviewers take into account when pulling the data, the funding sources of the randomized control trials. Yes,
okay. I mean, if I was just Joe Schmo,
I don't know if that has something to do. If the randomized control trial is well conducted, the funding sources should not matter. Shouldn't have an impact on the results of the trial.
So you think like publication bias isn't really an issue, and
publication bias might be an issue.
Okay,
we can address that in systematic reviews.
I mean, I'm just going to talk to you like a girl from Trucksville, Pennsylvania. Like, my mom always said, you don't bite the hand that feeds you. And it's kind of just, I mean, it doesn't raise suspicion at all. Because like for me personally, I'd like, well, if it was like sort of completely no conflict of interest, I would probably trust the results first. Dr. Kory, Pierre, do you want to comment on this at all?
Well, I almost feel like laughing at the idea that a study with 4 pharmaceutical sponsors whose interests run directly counter to ivermectin could be appeared as having not conflict of interest. But let's leave that aside for a second. I want to talk about the bigger problem, which is, and this is really a challenge, is I agree with Luis scientifically, right? That you want this trial, you want a definitive randomized controlled trial so you can prove causation, right? Or efficacy. The problem is those trials, the characteristics of trials, you need well-funded coordinators, investigators, statisticians, right? You need a good funding source, and you need infrastructure. And so the kind of trials that Luis is looking for are almost always for-profit medicines. When you look at the evidence base for ivermectin, which I call a nonprofit medicine, you're dealing with 60 trials, but the only one really that has conflict is the one that you just talked about. And it's the only one that's negative, and it's the only one in a major medical journal. Okay, so talk about publication bias. We can talk about reverse publication bias, which is the little guys don't get in. There's no conflicts of interest in this other evidence base. These are well-meaning clinicians from different centers and countries around the world. And this is the other thing I want to say, just use common sense. Leave alone all the methodology and all this, but the consistency of benefits from different countries, different centers, different investigators, all without conflicts of interest is truly remarkable. And this, when you have a system in which only what I call the big RCT, like capital B, big RCT, the only thing that will ever establish efficacy is a big RCT, and the only one who can do a big RCT is essentially big pharma, well, then you're always going to be stuck trying to prove the efficacy. And every time you try to bring evidence for a nonprofit medicine, you're going to be dismissed as not having rigorous evidence. And I think the system is bad. And so what you're left with is a system that produces almost like a monopoly pharmaceutically engineered, high profit medicines. And it's really bad. Look at remdesivir. That is, I mean, remdesivir is a joke. It's a joke how that thing is now being used around the world at $3,000 a dose. You know, here we're debating ivermectin. We really should spend an hour on remdesivir. How many billions are being made on a drug that has like almost no effect?
Okay, that could be another podcast.
Yeah,
I'll put it on. I'll put it on the schedule. I want to thank you guys both for commenting on that trial. So there are a couple. I wrote down 3 randomized controlled trials that look like they're in the works. I'm not exactly sure what stages they're at. There's one at the University of Minnesota, recently started. There's the Together trial at McMaster University. I believe that's also studying ivermectin. And then the NIH just launched the Active 6 trial, where they're going to study ivermectin, and they hope to get around 15,000 people at Duke University. Is that right? If I'm wrong, you can jump in, but
you got it right.
Okay, so should we wait for the results of these trials? I mean, these are going to be what would be traditionally considered more rigorous, or should we trust the results of these trials? Whoever wants to jump in first, go ahead.
I think this research that you mentioned are doing what it should be done. So the available evidence so far doesn't allow to make a recommendation, but it stands that we have a great field for research if they conduct that research well, and we can have those results near in the future, and we can trust that the risk of bias is low, or we can have a good appraisal of that research and those findings, that we may be able to provide a recommendation in the future. I think this is what we should be doing right now. If based on the available evidence so far, we cannot make any recommendation, at least for me, that's a personal opinion. So we should conduct more research. That is, it's the way to go.
Okay, Pierre, do you want to comment on that?
Yeah, so let me pick out that last statement. The fact that you can't make a recommendation based on this available evidence, I find completely ignores the history of recommendations. You know, we don't need a 1A recommendation. A 1A meaning on the highest, unassailable, largest randomized controlled trial evidence with high degree of statistical significance. That's called a 1A. That is a strong level of recommendation. All of the guideline agency, all of the public health agencies and guideline development groups have numerous recommendations available to them. You don't need a 1A. In a pandemic, on a safe drug, you can give a conditional recommendation. You can give a weak or a cautious one. You can collect data prospectively and then reassess. You are not going to harm people. We're in a pandemic. People are dying. Countries are being overrun. And we want to sit around and wait for 1A evidence. That's ridiculous. Number one. Number two, the meta-analysis of randomized controlled trials is considered the top of the pyramid, okay? And now he might object to some of the qualities of these systematic reviews. However, there are systematic reviews, one that is about 2 days from pre-publication, which has ascertained using multiple different forms of trial sequential analysis, rigorous detail of each of the composite trials, moderate certainty of evidence that it saves lives. And you're asking me what I think about 3 large prospective randomized controlled trials using placebo. When you have 60 trials of benefit, you have a meta-analysis done by veteran WHO researchers from the UK who've been doing consultant guideline development work for decades, who are saying that it's a life-saving drug. There's no way, it's ridiculous that I would ever condone a placebo at this point. The other thing that I want to say is I actually treat this disease. I'm at the bedside. I've been using ivermectin in multiple phases of disease, not only prevention, early and late treatment. I know this stuff works. Every doctor that I know who's incorporated into their practice knows this works. The diminution, mitigation of symptoms within 24 to 48 hours of starting it is reproducible and consistent. It's an effective drug. I can't understand. Everyone is obsessed with these big RCTs. You don't need the big RCTs to know that it's effective.
You don't need it for ivermectin, but you need it for other drugs.
Not at this point with ivermectin. You have dozens and dozens of trials, observational, randomized, all matching, many statistically significant. And in meta-analyses, you have massive benefit on mortality. Even a low certainty evidence, even though it's graded as moderate, even if you had low, an important outcome such as mortality, even at low certainty evidence, that is a very important finding, and it should be applied into clinical practice. Come on, man. I'm at the bedside of these patients. They're dying. They're crashing onto ventilators, and they're dying repeatedly. I can turn them around. I can keep them away from hospitals. Mexico is reporting that. Argentina is reporting that. India is reporting that. We know how to keep patients from hospitals, and yet we want to conduct these North American, high-degree, sophisticated trials where we give patients placebo. That's ridiculous. On that consent form, in those trials, I hope that every patient can read all of the benefits been showed in all of the trials, even the associations rather than causation. If I tell a patient, well, we have hundreds of thousands of patient data showing a very high degree of association with survival, it's not actually proof of efficacy, just association. I wonder what those patients would say.
Louis, are they using ivermectin in Argentina?
Yes, I believe that they are using off-label, of course.
We use a lot of things off-label, right?
Of course.
Yeah.
I believe there might be some institutions using ivermectin.
If a relative asked you for ivermectin, what would you say? If they really wanted it, they were really concerned, they were convinced by the evidence.
Go into a trial, take a placebo.
I will, first of all, I would like to tell Pierre first, I have no personal issues with ivermectin or anything. Right. The recommendations since the GRADE approach are based on strong or weak recommendations against or favor for the treatment. 1A, 2A, 2C, I don't care, but the recommendations are now trying we are trying to do the recommendations in following the GRADE approach. And the systematic reviews, they don't tell you what to do. They only assess the available evidence.
True.
And they also tell you what we do not know so far. And that is why I'm totally agree with those new trials that are being conducted.
Luis, I like the point that you mentioned that, right, that the systematic review just assesses the medical evidence. When you make a guideline recommendation, right, there's many more factors that are taken in. There's things like equity and costs and feasibility and safety, right? And so, and you think about it on a population-based level, and for instance, the Bird Group in the UK, they did a guideline development process consistent with the standard of WHO. They had 65 patient advocates, researchers, clinicians, and specialists from around the world who assessed that systematic review evidence and made a vote and a recommendation based on all of those metrics. And they overwhelmingly voted to a very strong degree that ivermectin should be deployed immediately in the prevention and treatment. And so when you follow that process in an independent fashion, you arrive at a very strong recommendation. Now, I'm just going to stop and say, if you want to talk about the WHO and what they did, because you mentioned that earlier in your opening remarks, I'm more than happy to comment on the WHO, because to reference them as a serious standard to follow as guidance around ivermectin is fraught with severe and dangerous consequences. They should not be listened to, and I have abundant evidence to show why they shouldn't be listened to.
Thank you for that. So we're running short on time. I just want to ask one more question, which I guess is also a philosophical question, if you will. So obviously the US is fine when it comes to vaccines. Right now, we have an abundance of supply. Our problem now is a stagnant demand. We're doing good, but other countries are not. There's many countries out there that are really, really struggling. They're not getting the vaccine and they're broke, they're poor, and their cases are going up, their hospitalizations are increasing. And many of the people are asking for ivermectin. Many of the doctors want to use it, but their hands are tied or they're following guidelines. And, a lot of people are actually going to the black market to get it. and let's go back to how much ivermectin cost. It's cheap, right? And Dr. Kory, you can correct me if I'm wrong, but I think it costs less than $2 for a 5-day course of ivermectin.
Depends where, but
okay.
yes.
So
yes,
it's
exactly.
super cheap. So and it has a good safety profile, as I think both of you agreed with that. It seems very safe. So Louis, what are your issues, or your would you say go ahead and use it if they want to use it? You're in the situation. Why would you say use it? Why would you say not use it? And we're talking about these countries now that are desperate, just taking it outside the US for a bit.
Yeah, well, I think I live in one of those countries you mentioned. Are kind of short on vaccination. We are getting better right now, but the fact that it's cheap and the fact that it's safe, but it's probably safe, doesn't mean that you have to use it for COVID. And I wouldn't recommend it yet. Based on the available evidence, the price or the safety is only one of the issues that I would look to make a recommendation, but I still lack the clinical benefit. And
so if do you see patients?
Yes, totally.
You do?
I
Okay.
work in an intensive care unit. I'm not a doctor, sorry, I didn't say. I'm a physical therapist. I specialize in critical care, in respiratory care and mechanical ventilation. So I see COVID patients all days.
So you would, so if people if patients asked you for in Argentina, no vaccines available, you would just say no.
As a physical therapist though, right? It's not as a physician, treating physician, navigating myriad, developing physiologic changes and co-infections, complications, dosing, titrations, all of that, right? It's physical therapy.
Yes, I'm in charge of mechanical ventilation, mainly in the ICU.
Okay, and I know there's different thing, in the US probably is different than Argentina. You would just say no, don't use it. Because until the World Health Organization says go ahead.
I haven't seen any recommendation on ivermectin. There's lots of uncertainty yet, so I wouldn't recommend it.
Okay. Pierre, do you want to comment on that?
Erin, right now, as we speak, there are 14 controlled trials on prevention. I've never seen benefits like in these 14. You have 14, all large magnitude, highly statistically significant associations, causations of ivermectin use with reduction in the risk of getting COVID to incredible degrees. When you look at from the Carvalho trials, where you paired it with Garegnani, the seaweed, 800 people who took it over 4 months, not one got sick, right? They took it weekly. You just had a study coming out of India where they studied healthcare workers, 3,200 of them. And those that took just 2 doses over the next month, a huge reduction, up to 83% reduction in transmission. It rivals the efficacy of vaccines. You have 8,000 patients in these observational and randomized trials. And yet a safe, cheap, widely available medicine, 14 trials, high degree of statistical significance, and yet we won't use it. Well, I'll give you my answer. I've been taking ivermectin weekly now for about 8 months, and that's what I'll continue to do. I haven't gotten COVID, N of 1, but I will say that's consistent with this large degree of trials evidence. And so I have
2 anecdotes, right? 2 anecdotes equals data.
Yeah, I have many people in my network who've been prophylaxing that I recommend prophylaxis for, and my group advocates for a prophylaxis protocol. So we are firmly convinced of its efficacy in reducing transmission. About the only question you should have is what is the magnitude of benefit, not that there is a benefit. That's unquestionable. You can't design 14 trials and find large magnitudes of benefit and somehow get it wrong. I'm sorry.
All trials
Not to that
assess
degree.
the same outcomes, the same doses, same
Transmission.
population.
It's
And
different
they were all-
populations. So post-exposure, same thing. High-risk workers, same thing. So you can pick the population. So if you have exposure to someone who's sick in the household, we have a number of trials showing that if you give it to the household members, massive reduction in those who actually contract COVID-19. If you take high-risk healthcare workers, that's probably the largest population, and that's a pretty good population because they have the highest risk of getting it, and you have massive reductions in their risks of getting COVID-19. In fact, for me, out of all of the different populations that we've talked about, I am most impressed with the prevention one. And I want to point out one very curious fact in the world that we really should address is why the WHO, in their guideline statement on ivermectin, they have one sentence, and this is how the sentence reads: "We did not review data on ivermectin in the prevention of COVID-19. They just decided not to. I wonder why. I haven't been able to figure out why they wouldn't want to look at a preventative for COVID.
I believe that issue is addressed in the Pan American Health Organization reviews. You can check that out.
As to why they didn't want to look at the data?
No, I believe the Pan American Health Organization is performing rapid reviews. No, not rapid reviews. Yes, I think it's rapid reviews, monthly basis with updates every month.
I was talking about the WHO, why they wouldn't want to review that.
I don't know what you should ask the WHO. I'm just saying that I believe that Pan American Health Organization has addressed that topic in their reviews that are updated every month.
And you know what they said? I actually don't know what they said.
I can guess. Can I guess?
Well, I mean, if he knows, that's probably better if he knows.
I would say it would be certainly not a supportive statement, and only for one reason: that the Pan American Health Organization is subject to the same external influences that WHO, which is sponsors and funders, as well as almost a near total commitment to a global vaccination policy. So ivermectin is inconsistent with carrying out that policy, and so I cannot imagine a supportive statement on this incredible evidence for prevention of ivermectin as a preventative. Would be no different than WHO. WHO went one more. They just don't even want to look at the data. They don't even want to look at the data.
All right. Well, you guys, I think we're out of time here, but I want to thank both of you. I think that was a very good discussion. I also learned a lot. And Louis, I hope your country, I know your country's been getting hit hard. I hope you get some vaccines from the good old USA. I think we promised to send some.
I believe, yes, I believe
yeah,
so, yes.
Yeah, hopefully we do. And Dr. Kory, I want to thank you for, in telling us all about your view. And I know you're really in this fight, and it's great to hear both sides. And if you guys want to offer closing words, go ahead, because my dog is about to bark and offer his wisdom on ivermectin if I don't go yet.
No, I think that it's ivermectin use in COVID. It's a field for research. I agree that it might have a potential benefit. I have my issues with this experience-based data that Pierre has exposed here today, but in order to make recommendations, we need better evidence. Just that.
Pierre, any closing words?
Yeah, no, I'll just say that in my career, I've been studying interventions, trying out different ways to treat multiple disease models as an intensivist and lung doctor. And I have never come across an evidence base that is so consistently positive from so many different sources. So from prevention, early, late treatment, observational, randomized, they're all saying the same thing. Observational, in fact, most disease models you study, there's always conflicting evidence. You'll have a trial that suggests that it works, one that doesn't, in a different time or different population. Here you have nothing but consistency. And then the epidemiologic data is also incredibly consistent. anytime you look at where ivermectin is being used or adopted in a guideline, it's followed by reduction in deaths and case count. So from trials, from basic science to trials, prevention, early, late, epidemiology, and then myself and a growing network of colleagues around the world who are using it and are screaming from the rooftops that it is an effective therapy, I can't debate this anymore. And this is why I'm so impassioned about it, because COVID is a treatable disease. And although there's multiple possible treatments out there, I think ivermectin is the mainstay, and it should be globally and systematically adopted, period.